Okay, so I guess since you guys came in from the sunshine and are here, we should probably get started so you can get back outside as soon as possible, right? So, yeah. Yeah, it's amazing how beautiful it is out today. Okay, so I just wanted to start by answering a question that somebody asked me at the very end of the last lecture. Hey. Yeah. Thanks. I just wanted to start by answering a question that I was asked at the end of the last lecture. So in the last lecture, I went through this biomechanical analysis of the ocular motor plant, right, which is, you know, the tissue surrounding the eye and the muscles, and basically the load that you have to activate in order to make an eye movement. And we did that from the point of view of trying to understand the forces that you would need to generate for a viscoelastic system. And then I went through this analysis that Dave Robinson, right, one of the founding fathers. Yes?
I'm sorry, I don't think the mic is working.
No?
No.
Maybe I didn't turn it on. That could be the problem. No, it seems like it's on. Can you hear me now? No? Is this one? Does that have a switch too? Can you hear me now? Yeah. Okay, I think let's try it. It may have just been upside down. Is that better? Yeah, okay. Thanks for letting me know. Okay, so as I said at the end of the last lecture, someone asked me a question. So the question was about how do we relate the mechanical analysis that I went through, where we went through Hooke's law and the viscoelastic model of the muscles, and then we talked about the inertia of the eye. How do you relate that to the actual motor neuron drive that Dave Robinson actually demonstrated when he went in and characterized those motor neurons and looked at the actual control signals that were being generated? And I went through it a little quickly. So the main point is, right, is that ideally what you'd want to do to make that link is measure eye movement and then torque, which is rotational force, right? Because that's what the eye muscles are generating. But we can't really do that. And I mentioned somebody's tried to do that, and you know, it might be worth reinvestigating as everybody's miniaturizing sensors. But what people did in the past was they actually measured firing rate, right, as a proxy, because you know, that tells you what kind of signals are coming in and activating the muscles. So you don't have a direct measurement of torque, but what you have is a measurement of the firing rate of the neuron that's driving that torque. And one of the things we discover in comparing those two analyses, right, where we went through building that biomechanical model of the ocular motor plant and actually looking at the neural drive, is that they have the same structure, right, that you end up pretty much simplifying to a first order differential equation, which has an elastic term and a viscosity term. And what that suggests is that the firing rate is pretty much proportional to the force that's generated, right? So that's kind of the link that you can make between those two analyses. So what I'm going to do today is go on to another system. When I initially talked about eye movements, I said that there are 5 kinds of eye movements, and that you had actually focused pretty much in your previous lectures on one type, which was saccadic eye movements. I also talked about other voluntary movements like pursuit and vergence movements. And then I said there are a couple very important reflex movements. One is the vestibular ocular reflex, and the other is the optokinetic response, and they help you keep stable gaze as you go through the world. So what I'm going to do is talk from here on in, I guess, about the vestibular system, which is a pretty fundamental system. People may have heard about it, you know, in high school biology, maybe since, but it's something that is really working all the time for us in the background, but that we know surprisingly little about. Like, we have a pretty good sense of the auditory system because you can plug your ears and know what it's like not to hear. And it's the same with vision. You know what it's like not to see. But it's really hard for us to appreciate what it would be like to go through the world without our vestibular system. So I'm going to start by just kind of explaining the function of this system, its basic organization, and what it does for us. And then I'm going to talk about the coding principles that are used in the system. It ends up being a really nice system for trying to understand some basic coding properties, both in terms of sort of traditional sensory approaches to looking at how the brain encodes information, but also with respect to how do we encode information when we're actively generating our own sensory input. And you'll find sort of a surprising answer to the second part. I want to note that this lecture looks pretty long. We probably won't finish it all today, so it'll probably be part of it run again on Monday, and then we'll go into the active lecture that I've posted up in Blackboard. Okay. So the vestibular system, the lecture that I'll give today is organized as follows. I'm just going to talk about the pathways and the basic function of the system. I'll then talk about linear systems approaches that have been used to analyze information transmission in this system. I'll then talk about recent experiments that have been done to look at the encoding of natural vestibular stimuli and what are they? What are we normally exposed to in our everyday lives? And then new evidence for nonlinear codes, including spike timing, and then the implications of this for designing things such as a vestibular prosthetic, which is a sort of a new innovation that people are currently working on actually at Johns Hopkins. Okay. So the vestibular system is a set of 5 organs on either side of your head that is situated in order to tell you how your head is moving through space. And this is a really important system. It tells you, gives you both rotational and translational motion information, and helps you do things that you take for granted, like keep your visual world stable on your retina as you walk down the street. It's also critical for things like postural reflexes. If you've ever slipped on ice, you find yourself catching your balance before you even realize what's happened. That's your vestibular system at work. And of course, it's also important for letting us do really complicated, maybe not all of us this complicated, behaviors that require keeping track of how you're moving through space and building what we call internal models. Okay. So what does the vestibular system look like? And this may be more familiar to some of you than others. So I think Xiaosheng talked about the cochlea and the auditory system, which is part of the inner ear. And then continuous with that, so if you guys remember, there's a scala media, which is the middle component of the cochlea, is filled with fluid called the endolymph. That's all continuous with the 5 end organs of the vestibular system. So that's what we can see here. So there are 3 looping canals, the anterior, posterior, and then the horizontal canal, kind of like bicycle inner tubes. They're all filled with fluid. And they each have a sort of a business end, which is a little bulge called the ampulla, and that's where the sensory receptor cells sit. And each of the semicircular canals comes in and back, or out and back into the utricle, which is a linear sensor. It senses linear motion. And then just below that sits the saccule, which is another linear sensor. So if you look at these 3 canals, what's kind of, I think, fascinating from a point of view of an engineer's design project is that they're pretty much organized at right angles to each other. So they're orthogonal. So by activating one versus the other, or some ratio, you can determine your 3-dimensional motion, rotational motion in space. And then likewise, on the utricle, you have a 2D map of linear motion in a plane that's parallel to the floor. And then there's another map like that, but that runs vertically in the saccule. So you have this 2D map and then another map, so you can sense 3-dimensional translational motion. And if you think of the MEM sensors that are now available, right, the utricle and the saccule are a bit like accelerometers, right? They really measure your linear acceleration through space. So if you actually tilt your head, they'll tell you how you're oriented relative to the g vector, right? Because that's a cue of linear acceleration. And then in addition, the rotational sensors are effectively like gyros, and they give you your rotational velocity. Okay. So, yeah?
How would this be effective in an anti-gravity or gravity?
How would it be so a lot of people who work in the question is, how would this be affected in anti-gravity? And that's a model for looking at the fact that the brain's going to have to sort of reconfigure its internal models of the sort of sensory inflow it expects when you move through the world. So when you're in 0G, you don't have that, gravity vector working anymore on the otoliths, so they end up not tethered in the same way, and there's basically a lot of central compensation that has to go on. And one of the thoughts of why people get quite space sick, which is not well advertised, but a real phenomena when astronauts go up the first day or so, they're pretty miserable, is because they have to basically sort of recompute what kind of information they're going to experience as they move through the world and what actually happens. Okay. So I'm going to just now mention 2, so vestibular pathways are important for the things we do every day. And there are a couple classic ones that people have studied a lot. And that's because they're so simple compared to the other things that the brain does. They're really beautiful model systems, even in mammals, right? So the first one is the vestibular ocular reflex pathway. You have vestibular information that goes into the vestibular nuclei and generates reflexes within 3 neurons. So you have a vestibular afferent, which comes through the 8th nerve, a shared nerve with the auditory system, goes into a central nucleus. Here, I've lost my pointer. I don't think that's working. There we go. Okay, it goes into a central nucleus called the vestibular nuclei. So that's the first central neuron. So you have cranial nerve, which is a sensory nerve, projects into the vestibular nuclei. And these nuclei neurons actually project directly to the motor neurons that move your eye. So last lecture I talked about abducens and the trochlear and the third nucleus. These neurons project directly to those motor neurons, so that the link between sensing your head motion and moving your eyes is basically a 3-neuron pathway. Okay, so that would be what we call this gaze stabilization reflex. We also have postural reflexes, which, as I mentioned, if you're walking and you slip on ice, you have a 3-neuron pathway that connects the vestibular system afferents down to the spinal cord, so you generate very rapid compensating postural responses. And then, of course, we also have the ability to sense our motion. So if you're standing on an elevator and you feel the elevator go up, you don't have any visual cues or anything else going on. What you really have is that sensory input from your vestibular system, and that's done via pathways up to places like thalamus. Okay. So what I'm going to do is now just, again, these are what I just sketched out, and here are some real names put on the different nuclei. So this is all the vestibular nucleus. There are 2 subgroups of neurons that really are devoted to one versus another type of processing. So I'll start with the VOR pathways. The VOR pathways are mediated by a group of neurons called position vestibular pause neurons, because they have vestibular signals, but they also have position signals, sort of like what we talked about on the motor neurons the other day. And they get input from the prepositus, which is probably important for keeping your eyes fixed after you've moved your head, right? So they show this sort of integrated signal that we talked about in terms of saccades. It's the same integrator. And then again, they project off directly to motor neurons to move the eye. And the idea is that as you move through the world and your head is moving one way, you want an eye movement in the equal and opposite direction to keep your gaze stable relative to space. And that's what this is doing for you. And then again, there's this other pathway that's involved in vestibular spinal reflexes. It's a different class of neurons that we call vestibular-only neurons. I'll talk a lot more about those in my next lecture. They're very interesting. And they project to neck motor neurons and other motor neurons deeper in the axial musculature through the spinal cord, and do things like maintain your balance and posture. And they also have ascending projections up to the thalamus to give you your sense of motion through space. So this is just to give you an example of what life would be like without a vestibular system. So my postdoc took this film back when I moved here recently from McGill. I was in McGill for the last many, many years, but I've been at Hopkins for the last couple of years. And this is the street walking home from work in Montreal. Just walking down the street, and you can see he's got a GoPro on his head, right? And it's kind of uncomfortable. It looks like one of those, arty movie shots, right, where everything's kind of bouncing around. That's not the way the world looks as you're walking through the world, right? And that's because instead of just having this eye fixed in your head as you walk, for every little movement you make, your vestibular system generating an equal and opposite eye movement to keep the world stable on your retina. And I can show you examples of this and how fast it is. It's probably one of the fastest things that the brain does for us. So this is red is a head movement, and yellow is an eye movement. This was done in monkeys, so we could use really high acceleration perturbations and really look at the latency. But what you can see here is the time between head movement and eye movement is about 5 milliseconds, right? That's super fast. And that's consistent with the pathway delays in that 3-neuron pathway that I just showed you. And it's very consistent. What is sort of interesting from an engineering point of view is when we did these experiments, it was funny, the VOR latency was getting shorter and shorter every year as people were coming up with more and more dynamic stimuli, because what you need is a big signal, right, before things rise out of the noise. So when we started our experiments, the VOR, or when I guess I started my PhD, the VOR latency was about 20 milliseconds, but by the time we actually had a really nice input to look at latency in a really crisp way, we realized it was much shorter. So again, this is consistent with both the pathway delays that I showed you for that 3-neuron arc, and also suggests that the activation times of the muscles themselves are super fast, right? Because it's about a millisecond per neuron in a 3-neuron arc, and then you've got another 2 milliseconds to get the muscle activated and generating torque. And just for comparison, how long does it take to make a visually generated eye movement? Do people remember? I said that on Wednesday. So between seeing a target, could be a pursuit target, and generating a smooth pursuit eye movement, what's the fastest we can do that? Does anybody remember? It's 90 milliseconds, right? So this is an order of magnitude faster. So why is that important? Because when we're walking through the world, we're generating really dynamic movements. And what happens is the VOR works very effectively for us at high frequencies. So this is a log scale. So you can see at higher frequencies, the gain is close to 1. That means you get the same eye movement for the head movement you generate. So you really have good stable gaze. At lower frequencies, for reasons I'll go through as I talk about how the sensor actually works, your compensation would not be so good if you only had a vestibulo-ocular reflex due to the way that the organs are organized. And that's where the optokinetic reflex comes in. So normally we have multi-sensory integration, right, at work as we're going through the world. We have the visual flow, let's call it optic flow. So if anyone's been ever sitting on a train and the neighboring train starts to move, right, you get that big optic flow and you actually feel a sense of self-motion. So that can give us this self-motion cue. And that works very effectively at low frequencies. At higher frequencies, because the latencies are so long, it's a very sluggish system, it doesn't work very well at all. So you can see it's down at a pretty miserable gain below, what is it, well below 0.1 at 1 hertz, right? Just for reference, our normal locomotion frequency is somewhere between 2 and 3 hertz, right? So you really need this system. Okay. And this just shows another video to make the point that the vestibular system is also important in posture. So I'll just show that now. Oops. If it works. Looks like it's not going to work. Oh, okay. So this is a person who's just had bilateral vestibular loss, and he's asking to walk sort of heel-toe, like somebody might, standard test that a police might ask somebody who's been pulled aside for looking like they're driving under the influence. And he looks like he's having real difficulty doing that, right? It's just keeping his balance is really non-trivial. He doesn't have these compensatory reflexes built in that would be normally helping him. Okay. And I'll just put a note that, again, we think about people being blind or people being deaf, but vestibular loss is a real problem. In particular, if you do have vestibular loss, which you could have as a function of a number of different, it could be something that's genetic, it could be use of certain ototoxic drugs like gentamicin, certain viruses, their cerebellar syndromes will actually cause vestibular issues. And a really sort of, I guess, important thing for society now is that as you age, of course, we all know that people lose their auditory sense, right? Because the hair cells start to die or degenerate in the auditory component of the ear. But the same phenomena occurs for the vestibular cells. So that's part of the reason we think that people end up with poorer and poorer balance, in addition to, of course, other things going on in their muscles and their basic body structure. But the fact that you're losing peripheral vestibular input year after year after year is an issue. And one of the things you can do is upweight your use of that signal by doing certain types of training, which is what the clinicians are now working more on. Okay. So the way the rest of this lecture is going to be, so that was really an introduction just to motivate the importance of the vestibular system, so it's not just some sort of obscure system. And now what I'm going to do is talk about focusing primarily on the canals rather than the otoliths, because we know a lot more about the canals, in part because it's easier to rotate animals or people on a rotating device than it is to actually translate them. So But a lot of what I'll tell you for the canals, you can sort of apply to the otoliths as well. Okay. So the basic geometry of the canals is already mentioned. You have these three canals that are basically at right angles to each other. And they're nicely actually aligned with the pulling directions of the muscles that I talked about on Wednesday. The horizontal canal, right here, the way you guys are all sitting in class, it's probably pretty much parallel to the floor. And so your normal resting head position. And the superior and the posterior come off 45 degrees off the sagittal plane so that they're basically at 90 degree angles to each other. And as I already mentioned, each canal consists of a fluid path that comes in and out of the utricle. And then there's this sort of little bulge here. On one side, the bulge actually is called the ampulla and has the hair cells inside of it. So the hair cells, so here we're going to blow up one of these ampulla. The hair cells sit in this structure. It looks kind of mushroom-shaped. Again, the swelling is called the ampulla. The actual sensory epithelia where the hair cells sit is called the crista. And if we zoom in on the crista, you can see there are all these little hair cells, and they're all kind of organized so that the hairs on top of them, also called stereocilia, are organized with the same polarity. And if you zoom in on these hairs, you'll see that one is really tall, and then they come off in a graded fashion to be shorter and shorter. So they're all sort of lined up so that they are maximally activated by the direction the fluid would move through the canal. So in the canals, they're all lined up with the same polarity. The one difference in the otoliths is you have this beautiful map of the polarity of these hair cell bundles so that they make a really beautiful two-dimensional map of linear space. Okay. So the way that you activate the system in your everyday life is as you're moving through the world. If you shake your head yes or no, whatever you do that makes your head move relative to space, is as you move your head, the fluid is left behind in the canals. It lags a bit. And this is because it's got viscosity and also because the ampulla has some elasticity. This causes a relative motion between the ampulla here, right, and the fluid around it, because the fluid tends to lag, which basically pushes on the ampulla and bends the hairs or the cilia on top of the hair cells. Okay. So just to say it again, so the cupula is deflected by the movement of the endolymph, which occurs during head movements. Head turns, endolymph tends to remain stationary due to these forces, and then you tend to have the endolymph move relative to the canal in the opposite direction to the way that the head moved. Okay. So if you look at the vestibular system, in particular the canal part, the stimulus is angular acceleration or rotational acceleration. But over the frequency range that we normally generate head movements, the canal size in humans and other animals is very small. It's about 0.3 millimeters. And the fluid is more like honey than water, so it's highly viscous. And what this effectively does is produce an integration, so that over the normal range of head movements that we generate, the system functions more as a speedometer than an accelerometer. So the hair cell output, and in turn the afferents, are coding angular velocity rather than angular acceleration for the range of frequencies that we normally generate. Okay. So I'll go through that in more detail. And again, if you combine the information that you get from all three canals, you have this readout of rotational head motion relative to space in 3D. Okay, so remember we put this equation together to describe the motor neurons on Wednesday, which really simplified to this viscosity, velocity related term and an eye position term and a resting rate, firing rate. So people went through the same kind of analyses based on sort of the fluid dynamics of how the endolymph would move through the canals and came up with this formulation here, where you have a double derivative here, where this is the endolymph, okay, and then this term is the effective moment of inertia of the endolymph. Here we have the viscosity term. So this is an acceleration term, this is a velocity term, and then this is the position term, right, which would be the elasticity related term. Okay, And the idea is that you have this, now it's basically a second order differential equation, right? Because this term is also now present. And what this means is that when you solve that equation, you're going to end up with 2 time constants rather than just 1 time constant. And that's what's shown down here. You have one longer time constant, which is this viscosity term coefficient over the elasticity term coefficient. And then you have a shorter one, which is this first term, which is related to the moment of inertia over the viscosity term. Okay. So if you go back to our basic mechanical analysis, this is pretty comparable, right, to this, this model that we came up with, right, where you have inelasticity, viscosity that's shared, and then we have this additional mass, because here inertia actually does matter. Okay. And so I've assigned a homework, which I think you guys will get right after this lecture. I think there's a part A and B, which I will be for the two, basically equivalent to the 2 homeworks, which I think are supposed to be part of my section, where we take these sorts of formulations and map them into the Laplace domain, which I think most of you have been exposed to. It's a pretty easy homework. A lot of the coding's already been done as far as setting up the boxes and things in Simulink. Who's worked with Simulink already? Almost everybody? Who hasn't worked with Simulink? Few people? Okay. So those who have not, talk to those who have, right? But I think it should be relatively straightforward. But you'll have the opportunity to build some models based on mapping these second order differential equations into the Laplace domain to try to build different parts of both the afferent part of the vestibular system and then link it into other boxes that basically will generate a VOR for you. Okay. All right, so effectively that equation, just to put it into really plain speak, what it tells us is that the moment of inertia, the movement of the endolymph is opposed by 2 frictional forces, one which is due to the viscosity of the endolymph and a second which is due to the elasticity of the cupula, and that these 2 forces cause the endolymph relative to the cupula to lag head acceleration, right? Relative to what would be the case if only the inertia of the endolymph was significant. So the receptor cells which deflect the movement of the cupula are primarily sensed to head velocity rather than acceleration, again, over this range of movements we normally generate. So this is what I'm going to show you in the next slide. So I guess most of you have seen Bode plots, hopefully, at this point. So this is, and these are log axes on the x-axis, and then you have basically phase and gain up here. And what you can see is that we've broken this graph into 3 parts. There's the low frequency range, the mid frequency range, and the high frequency range. And the dotted lines indicate the range over which the system is transmitting velocity information based on the prediction of how fluid is moving through the canal. And this model is referred to as the torsion pendulum model. It's just a very, it's not a model based on recording neurons. It's a model based on how the fluid is actually would move through the canal based on its, fluid properties. Okay, or mechanical properties. So here is the middle range, and you can see phase here. Just make a note. Phase is 0. The convention here is phase of 0 is in phase with velocity. Okay, or head velocity. And then here you have minus 90 and plus 90. This is minus 90 here is acceleration, which is maybe a little confusing. It's just the convention they use for the graph, and plus 90 is position. So what this means is that at really low frequencies, actually the system is coding acceleration. At really high frequencies, you have so much lag that you actually start to have the system encoding position. And then the other thing is that what you can see is the system seems to be tuned for maximum gain over that mid-frequency range, which corresponds to the range over which we generate most of our movements during our everyday lives. Okay. So then there's another part of the exercise when you guys do your homework, which I suggest you try because it kind of helps link this sine wave analysis to really understanding the dynamics of things. The question is, what would you predict would be the output of this equation for a step input of head velocity? And what you can do is look at that in the next slide. And what people actually did is go in and record from the actual afferents themselves to see what they were encoding. And what you can see is they encode something that's consistent with what the torsion pendulum model would predict. Because you have this time constant, there's a really quick one that's so fast you don't see it, and then this longer one that corresponds pretty much to the time constant predicted by people analyzing the fluid dynamics. So you end up with this second time constant of about 5 seconds. The shorter one you can't see, it's just too fast. Okay? And these are two, so again, here we're looking at actual neurons, not model predictions. And this was totally predicted based on people's analyses ahead of time of just the mechanics of the end organ, which I think is pretty cool. Okay, so what you can see, I guess the thing to really take home from this slide, right, is here's velocity. It's constant. So that animal, probably a chinchilla, is zipping around at 100 degrees per second, but there's no modulation in the neurons, right? So what that means is that the system acts like a rotational velocity sensor when you have dynamic stimuli between, point 1 and point, or 50, whatever. If we go back here, I don't want to give you different numbers and then have everybody panicked. Right, so we'll say 0.03-ish and 50, right, hertz. But if you have something in steady state, which is basically the 0 on that x-axis, what you can see is you get a nice initial dynamic response, but then you're down to nothing, right? Because again, the system's really is sensitive to acceleration. Okay. So now I'm going to talk about, let's just go back up here. The prevailing view of analysis in this system was initially looking at using linear systems analysis and looking at rate coding, a lot like what I already went over for the ocular motor system. And it was a very dominant paradigm in the field for quite a long time. Then I'm going to talk about recent experiments actually that have been, I guess, expedited thanks to these small MEMS sensors, like these accelerometers and gyros that you can put on an animal or on a person and see how they actually move through the world. And then we'll talk about evidence for nonlinear codes, including spike timing. And again, I'll talk about the prosthetic at the end. So we'll see. I'll probably get through the linear part and a little bit of the first part of the nonlinear coding. Okay, so the vestibular periphery, hair cells, and afferent nerve fibers. So I already showed you the ampulla and the crista of the canal. So if we zoom in even further and we look and see what those hair cells look like, I told you that they have little bundles of hairs on top, and they have a real polarity where you have the tallest one, and then they basically go off in a graded fashion to become shorter and shorter. And if you look at the cell bodies, they look a little different, right? So you have one kind of cell body, which is very cylindrical and has pretty conventional synapses with the afferents, and that's called a type 2 hair cell. And you find those across all species. Those are seen in everything from fish to frogs to people. When we sort of crawled out of the primordial soup from the sea to land and became sort of land-based animals, that was accompanied by the development of a second type of hair cell called a type 1 hair cell. And it's a very interesting, I guess, synapse from the point of view of neuroscience. There are not many other synapses like this in the brain. So the cell itself is kind of a vase shape. People say it's sort of a calyx-shaped cell body. And then it has this type of synapse that comes up and hugs the whole cell body. So it's quite different than the type 2 hair cell synapse. And people are still trying to understand how synaptic transmission occurs at this hair cell. We know that there's some conventional transmission using conventional neurotransmitters, but there's also this idea of what's called effect transmission, where you build up local gradients and differences in potential that can actually be used in signaling, because you have this very unusual sort of process that engulfs the entire cell body. Okay. But what is clear when you look at these type 1 hair cells and this unusual synapse is that nature went to a lot of trouble to design this when we basically moved on to land. Okay. And I already mentioned this very briefly, but so in order to activate the hair cells, what you do is, just imagine we're going to record from an afferent and an animal whose not moving in space. Okay, so we have our electrode down in this yellow afferent. And if we could record the receptor potential at the same time of the actual hair cell, we would do that too. That's kind of hard to do in an attacked animal. But what we would see is if you moved in a way that pushed the hair cell bundle towards the tallest of the hairs, what that would do is depolarize the hair cell. So this looks a lot like the auditory system. And then if you push the bundle in the opposite direction, you would hyperpolarize the hair cell. And then what we can do is go down and see what that would do as far as the afferent that's connected to it and bringing its information that it detected into the brain. And what you can see is at rest, the afferent fibers actually produce a really high firing rate. They're buzzing away at about 100 spikes per second. They're really busy. And then if you depolarize the hair cell, they are activated. And if you depolarize, they're inhibited. So you're basically sculpting both directions of movement around a resting discharge. So what they do is sense change in motion, right? With these nice resting discharges that you can then sense both movements one direction and movements the other. That's really important for compensation. Let's say you have something happen. You have a small tumor on the nerve on your right side, which is not uncommon, and someone has to go and do a surgery, so you've lost function on your right side. You actually still have the left side, which will tell you how your head is moving in both directions. It's not going to be as good as having both sides, but it allows a lot of compensation. Okay. So the other point I want to make is if we then go and record from these two classes of afferents, as I told you, their firing rates are pretty high for resting rates in the nervous system. They're 100 spikes per second. But there are differences. So there may be 100 spikes per second, but the way those action potentials are played out differs. So the ones that are hooked, the afferents that basically bring information in from the type 2 hair cells fire at pretty periodic intervals. So if you looked at the inner spike interval of this afferent, you just have a nice spike, given that it's firing pretty much every 10 milliseconds, right? So you'd have a nice spike that every inner spike interval is pretty close to 10 milliseconds. In contrast, these other afferents that are hooked up to the type 2 hair cells are very irregular in their discharge. So not surprisingly, people called these regular afferents and these irregular afferents. Okay, so how do these neurons actually encode motion? And again, this is, traditionally what people have just done is use transfer functions and assume that the system was linear. They kept everything small as far as ranges of motion and would place sine waves in, so different sine wave frequency, and then record the output of the neurons and look and see, what's the change in gain. The gain would be in terms of spikes per second per degrees per second, right? That would tell you how much spiking you're getting for your input. So your input is degrees per second of motion. The firing rate is spikes per second. So your sensitivity is spikes per second per degree per second. And then you can also look at shifts in frequency, right? In phase. You can see as a function of frequency what's happening to the phase. Is the phase remaining constant? Is it moving in some way? And that's been really the way people have looked at this for years. So when you can do this, you can record in animals, right? From single fibers, either regular or afferent fibers, and then you can play in sinusoidal sine waves. So you'd have the animal, like a chinchilla or a monkey, you'd be rotating it back and forth at different frequencies and recording the afferent responses. And when you do that, oops. Okay, when you do that, this would be, say, 0.5 hertz. And then 15 hertz, right? But the same amplitude, so let's say both cases, let's say it's about 40 degrees per second peak velocity. What you can see is the regular and irregulars respond really nicely. So this is recording from single afferents. They're really beautiful. They give you an online readout of how your head is rotating in space. And they look very much like velocity. And what you can see, though, for the same velocity input, as you increase the frequency, you get a lot more modulation, right? So that's gain. So you can see here the gain of both the irregulars and the regulars increases as a function of frequency. And this is pretty much plotted over the range of head motion that we know is pretty much the physiological range. Oops. And then the same thing you can do looking at changes in phase. So you can say, okay, here's the head velocity, here's the peak of the firing rate. here is 0.5 hertz, here is 15 hertz. And what you see is that as you increase frequency, you end up with a bigger and bigger phase lead. So things tend to move towards acceleration. Right? You tend to lead the actual velocity stimulus when you look at these afferents. Okay, and when I ask you to work through the problem or the homework set, this is actually an important slide to think about. So so maybe just make a little note that you might want to think about the relevance of this when you're trying to build these block diagrams of how the system works. Okay. So this is the classic point of view that I just went through. Neural coding in the vestibular system is inherently linear. That's been pretty much, there are a lot of people who stubbornly hold on to this idea. That is supported by electrophysiology studies that have kept things in this very small range. And that the VOR is linear. And again, if you look at the vestibulo-ocular reflex and the eye movements and you don't move things with too large amplitudes or too high frequencies, it actually does look quite linear. Okay. But things to point out. Focus on gain and phase does not account for the signal noise. You can have a big gain, right, but could have a really big band of noise around that gain. And the sensory input that people have studied have been really low amplitude, usually about 20 degrees to 40 degrees per second, 0.1 to 0.2 Gs of motion. The question is, how do we really move in everyday life? Okay. Oh, and this is just supposed to make the point again that you have this variability in discharge for the regular and the irregular afferents, and you can see this difference in this inner spike interval, right, where the regulars are very much fixed on about 10 milliseconds, and you see this broader spread of ISIs, inner spike intervals, for the irregulars. So the idea is, because this is so much noisier, what you can do is a power spectra of that resting discharge. If you do that for the regular afferent, you see this, the sort of pinkish bar here represents your physiologically relevant range of head motion, so maybe up to about 20 hertz. So you get, now there's very little noise down here, and then you get these sort of oscillations. So you have one at 100 hertz, that makes sense, right? Because that's basically the firing frequency. Then you have the harmonic at 200 and 300, right? Because it's a very periodic firing neuron. In contrast, if you do the same analysis in the spectral domain, right, frequency domain, for the irregular afferent, you see all this noise down here, right, compared to the regular neuron, which suggests that you might have more noise in the physiological range, relevant range. Okay. So in order to really look at that, what people started to do is record from both types of neurons, but using different kinds of stimuli and looking at things like information coding and computing information metrics, and also looking at neural detection thresholds. So I'll take you through that, and then I'll probably stop at the end of this section. Okay. So now, instead of using just sine waves, you take advantage of a broadband noise stimulus that covers the range of head motion that we normally generate, right? So that would be, say, 0 to 20 hertz. We know, based on work done for labs like my own lab. And then you can record what's actually coming out of the afferents and see how is that type of information actually being encoded. And this shows a head velocity, a snippet, right, of one of those protocols. And this is the stimulus on top, head velocity. And here's the firing rate. Here is the actual spike train, right? So what we're doing is we're logging whether or not there was an action potential generated. So this tells you when the action potentials occur. In order to get this firing rate, what we just did is you convolved each of these action potentials with a Gaussian, right? And what you can just see by eye is that's a pretty good representation of what the head was doing, right? You can just see that. So if you wanted to try to know how your head was moving through space, and you didn't have a gyro on the person's head, you could, but you were recording from the afferent, you would have a pretty good sense of how things were moving through space. So it's a really good representation of head motion. And what you can do is, you can compute the basically do a stimulus reconstruction where you determine the optimal linear filter that you can convolve with this spike train to reproduce a stimulus. So, so maybe something even better than our just basic Gaussian convolution. And you can actually do a really good prediction of the stimulus based on the spike train, right? So this neuron, the coding fraction was 0.53. So you might guess, since I've set up this paradigm where we had regulars and irregular afferents, that we see something different with the irregular afferents. And indeed you do. So what you can see here, again, here's the head motion, here's the firing rate. If you looked at one compared to the other, they don't actually look that similar. And if you estimate your optimal linear filter and do a convolution, you don't actually do a particularly good job of reproducing the irregular firing rate, right? Now this is sort of shocking given that nature went to a lot of trouble to evolve this second stream, right? This is the more recently developed stream. So the question is, what is it doing? Okay. So as I just mentioned, regular afferents have better coding fractions than irregular. So this suggests that you probably also have better information coding. So what you can do is also compute another metric called mutual information. And what this shows is that here across this range, again, so say the physiologically relevant range is from about 0 up to 20, irregulars actually have pretty poor information coding, whereas the regular afferents do a lot better. And in contrast, if we looked at their gain curves, right, which I kind of blew past that maybe a little too quickly when I went through it initially, irregulars have much higher gain across the same frequency range than regulars. So the irregulars are higher gain, but they're also lower information. Okay. So what does this mean for motion perception thresholds? So there you can go off. There are a number of places with pretty elaborate ways to test human perceptual thresholds. It's important if you're trying to do, for example, mapping of motion from someone who's trying to learn to drive a particular new plane or some new helicopter, some vehicle that's complex and has certain types of dynamic responses to steering. And what you can do is try to map the motion they'd actually be experiencing into motion in these sort of constrained robotic ways. And the mappings get quite sophisticated and can actually give you a really profound sense of motion. So there's a place actually in Tübingen, the Max Planck, that's got this really elaborate, I don't know if you can see how big a person is, but this is a staircase, right? So it's like the ultimate carnival ride. And it's like a little ski lift compartment up here that you get into, and it sort of zooms you around, and it's got multiple axes. There's one up here, there's a track down here, where they can test human perceptual thresholds. You can also do this in a slightly more modest way in a clinic, just by rotating somebody around. And when you do that, what you can do is measure the ability of people to tell if they're moving to the left or right by pressing a button, or left, right, and get it down to the smallest thresholds possible. And then what you can do is compare those thresholds to how well you can actually discern left versus right when you actually look at neuronal responses. So to do that in this system, what you can just do from some of the data I've already shown you is, so you have your table moving back and forth sinusoidally. You record the neuron firing rate. This is highly schematic because it's perfectly clean, but it gives you the idea. And then what you can do is say, okay, at 0 velocity, I have a certain distribution of firing rate, which I'm going to plot here. And then what I can do is move to different velocities and plot those distributions and see where I can discriminate my two distributions. And what you come up with is typically in the literature, a D prime measurement. And people usually use D prime 1 as the threshold, which is basically the difference in the means over the square root of the sum of the squares of the variance over 2, right? And then when you compute your D prime for, say, a regular versus irregular, and this is just shows firing rate as a function of head velocity for a regular versus an irregular, you can guess as you cut through 0, right, that the distribution for the regular will be much tighter than the irregular. So you can predict already that regular neurons are going to have much better discriminability. And that's what's shown here. So you can see if you look at regular afferents, they can discriminate about maybe at best between 2 and 3 degrees per second of motion, whereas the irregulars have much higher thresholds. You need a lot more motion to know if you're moving one way or the other. And then this just sort of links it to what we know about human perception. Those numbers are from monkeys, but people have done a lot of work in humans. And the human perceptual thresholds, which are like monkey perceptual thresholds, are way down here. So what it suggests is that we need to pool across multiple neurons to actually build our perception of motion. Okay. So as I mentioned, traditional analyses focused on small amplitudes and didn't only considered gain and phase. They didn't really think about signal noise. And they were also very low amplitude. So I think I'll just introduce this one slide. So as I mentioned, people have now been doing more and more with the small MEMS sensors that you can put on a small chip, right? So you have these 6-dimensional movement recording sensors that will tell you your 3-dimensional motion in space, both linear and rotational. This, what you can do is put these sensors, this is an older version, this was from ST Microelectronics that we actually mounted in a little module when I was up in Montreal. There's a Sharpie pen for comparison. Everything's been miniaturized much further since then. But they were small enough that you could put them on people and have them run through a park, play soccer. You could also put them on a monkey's head, and we even miniaturized it enough to record mouse head movements. Because when you're doing all this neurophysiology, you want to know what is the sensory input that the system is experiencing and trying to optimize the encoding of, right? In order to know the strategy of what would be the best way to encode a stimulus, you really need to know what it is to start. Okay. So this is basically just a schematic of how the sensor would sit on the head. Here are the canals. So again, we have basically 3 dimensions of rotation and translation. And this would be what the data set might look like coming in from one dimension of that sensor. And what you can do is then take the orientation of the sensor and project its rotational axes, for example, if it's the gyro part, onto the actual rotational plane of the 3 semicircular canals, so you know what kind of input is going to the horizontal canal versus the superior canal versus the posterior canal. So you can basically do those projections. And what you can then look at is the distribution of movement, for example. So this is amplitude on the y-axis, time on the x-axis. Here's the distribution of velocities. And what you can see there are these big tails going out, up to hundreds of degrees per second, well outside of the range of what people normally test in the laboratory. And what you can do is something where you plot a log-log relationship between the power at a different frequency and the actual frequency. Across lots of sensory systems, people find what's called a power law, which would be a straight line relationship for this particular plot. Here's an example from the visual system of what visual scenes look like. And what we find, the vestibular system, it doesn't follow a power law. You actually have this break. There's a low frequency component and then a higher frequency component. Here it's fit by 2 lines. It's probably maybe better fit by an exponential, but it doesn't seem to follow a power law. And I think some of what people have done in the auditory and visual systems needs to be sort of redone, because the argument is always just looking at visual scenes, but outside of the context of what actually is hitting the retina, or outside of the context of what actually is being transmitted to the inner ear versus the 3 ossicles. So I think there's still a lot of work to be done on what the actual sensory inputs are to those other systems too. We know that these dynamics or this relationship of these natural statistics is altered by the fact that you're generating natural motion and the fact that you've got body mechanics that are filtering your motion as you go through the world. Okay, so I'll just summarize what we know about natural statistics, and then I'll begin with that on Monday. So we know that in contrast to what people have been focusing on in the laboratory for the last many years, you can reach huge, just during your everyday life, you can generate movements of 8G, right? I said before, most testing's down around 0.1, 0.2. That shouldn't be 200 degrees per second. It should be more like 500 to 600 to 700. That's a typo, but very, very rapid movements. They're highly non-Gaussian. They deviate from scale invariance, as I just showed you. And the question is, how do the vestibular neurons encode these? And that's where we'll turn to the monkey physiology on Monday. So I'll thank you for your attention, and happy to take any questions.